great opportunity, if you can get one, to get into a lab and do some research. And it's nice because it is a program, like I said, that basically is available in any of the departments across sort of the science and technology parts of the university.
Hi, welcome to the abstract presented to you by the Western Student Research Conference. My name is Janica, and I'm the creative lead for the abstract.
My name is Ananya. I'm part of the outreach committee for Western Student Research Conference.
And I'm Colette, and I'm part of the research committee for the program.
And we have a very special guest here with us today. He focuses on the immunology field, and his work has earned him amazing awards such as the Governor General's Gold Medal for Academic Achievement. He currently focuses on looking into the function of phagocytes in removing apoptotic cells and how failures within these actions can lead to diseases such as atherosclerosis, among others. He also studies phagocyte biology, but obviously he has done some amazing work in other parts of immunology as well. Everyone, please welcome Dr. Bryan Heit.
Thank you for having me.
We're so glad you're here. So we're going to get more into, your background with research. So you have been involved with immunology research for a while now. So would you like to just elaborate on why you sort of wanted to get involved with immunology research?
I started actually as an undergrad student quite a while ago. And my sort of interest was sparked when I took my first immunology course. And I took sort of back-to-back an immunology course and a virology course and really became interested in sort of host pathogen interactions. And so in my fourth year of undergrad, I did an honors degree where I spent a year doing research in a lab at the University of Calgary, where I did my undergrad. Where I began to investigate a type of immune cell called a neutrophil, which is one of the main sort of immune cells that remove bacteria during infection, and really trying to understand sort of how they decode the different signals they get inside of an infection, the site of an infection, to sort of figure out how to migrate towards the bacteria and remove them while ignoring some of the other signs they see of tissue damage and things like that. Following that, I actually continued in the same lab for my Ph.D., sort of pursuing that same project, as well as doing little side projects that probe things like HIV, Staph aureus, and a few other infectious diseases that sort of impact on that same pathway. From there, I went and did what's called a postdoc in the Hospital for Sick Children in Toronto, where I stuck with the theme of immune cells, but I shifted from trying to understand sort of immune responses to actually looking at sort of the fine cellular level activities of immune cells and how some of their functions are regulated at a cellular and signaling level. Really investigating a process called phagocytosis, which is where immune cells will engulf and then kill, bacteria and things like that. And really trying to understand some of the signaling and the other processes that let that work. And so following that, I came here to Western. I established my lab and sort of continued on with that sort of phagocytosis-centric theme. So we're very interested in phagocytosis and a special form of phagocytosis, which is called epherocytosis. And the difference being phagocytosis is eating a pathogen, epherocytosis is eating a dying cell. And sort of phenotypically, they're very similar. A macrophage or another immune cell comes along and engulfs either the pathogen or the apoptotic cell and breaks it down. But immunologically, they're very different because, of course, pathogens are hazardous things that you need to get out of your body, whereas dying cells are very common. We make something like 100 billion of them every day just through turnover of cells and minor injuries and things like that. And obviously you don't want to be launching a huge immune response when you're basically cleaning up just the day-to-day mess that forms in our body. And so we know that the epherocytosis process often goes wrong, and that's where some diseases come from, or it can help drive some diseases. So our interests kind of fall into two camps. So I think you mentioned atherosclerosis at the beginning, so that's our major focus. for people who don't know, atherosclerosis is why we worry about cholesterol. And so essentially you get cholesterol depositing in the blood vessels of the heart. Normally macrophages remove those and clean them up, but for some reason, actually in about half of us, that process eventually breaks down and the macrophages begin to just accumulate fats and cholesterol. And they turn into what's called a foam cell, and it literally looks like a mass of bubbles because there's just so many little droplets of fat inside of these cells that there's not much else to them. Those cells eventually die, and if they're not removed, they begin to accumulate and form a plaque. And the plaque is what we really worry about because that can occlude blood vessels in the heart, or if it ruptures, it can cause a heart attack or a stroke. And that removal is through efferocytosis. We recently discovered that efferocytosis is probably the first macrophage cellular activity that fails during the onset of disease. And there's been other groups that have actually shown that if you can help sort of reverse that defect, that you can actually reverse some of the damage that occurs. And then the other area we're interested in sort of spans both pathogen phagocytosis, but also autoimmunity. And we're trying to understand how it is that a macrophage knows when it's eaten something hazardous that it needs to trigger an immune response, and how does it go about doing that? But also, how does it sometimes mess up and trigger an immune response when all it's removed is an apoptotic cell? And so that's sort of the other theme in our lab.
So you've done some pretty incredible work over the years to develop into the great scientist that you are today. What kind of drives and has helped you develop the questions you ask in your research?
I mean, a lot of them kind of produce themselves, so to speak. Half the time when you answer one question, you just create 10 new ones that are needed to be answered then. So a lot of the work we're doing now really came out of unanswered questions that sort of arose in my postdoc when I was working at Sick Kids. Where we investigated a protein called CD36, which is the receptor that takes up bad cholesterol, but it also can take up apoptotic cells as well as malaria. And somehow macrophages knew how to sort these different things apart, even though they were using the same receptor to take them up. And so that's kind of where I got on this track. And then it seems every time, we discover how one part of it works, it just leads to 3 or 4 new pathways that we have to kind of decode and figure out to understand deeper how these things are functioning.
Okay, that's really interesting to hear. So you bring on some undergraduates into your lab as well. And is there anything you wish undergraduates were more aware of prior to getting involved in research?
Actually, I find the undergrads that come into the lab are pretty well prepared for research. I think what a lot of undergrads maybe don't appreciate is, one is the number of opportunities there are for them to engage in research, and then how to go about finding those opportunities. There's always a rush right about this time of year, actually, of students asking for summer student positions and to apply to some of the funding programs for that and stuff. And unfortunately, you're too late if you're asking now. We've usually matched those positions in the late end of the fall term. And so, students, I think, need to be a little more proactive sometimes than they are and reach out to professors at the beginning of the fall term rather than, waiting until those summer student positions come up. There's a wonderful work study program here at Western that can act as a supplement for students who get government scholarships. And that's another great entry point into a lab, and even better, you get paid to do that. It's funny because we post positions, work study positions for the lab fairly frequently, and I'd say about half of them go unfilled, even though I know that there's students out there looking for those types of positions and who would qualify. So I think that being a little more proactive and trying to find these positions a little bit earlier in the year, and also being aware of all the programs that are available are really what I think students need to maybe do a little bit more of in order to get those research opportunities.
Well, thank you for pointing out some of the starting points for students who don't know where to begin. I also wanted to ask you that in your experience, the pandemic is about 11 months old now. And so has student experience or the process of applying for research positions changed in any way?
Absolutely. As you can imagine, with the outbreak, with the extra sort of security layers that we have to deal with in the labs, it's hard to bring someone in, especially a junior person, because even training someone now is 10 times harder than it was before the pandemic. And obviously the university is sort of restricting who can come into labs. And unfortunately, with sort of the exception of honor students, so fourth year students working on an honors project, most undergrads aren't really able to come into the lab at the moment. And so, we've gone about doing our normal summer student recruitment like we usually do, but we've had to tell, everyone that's applying, like, I'm happy to apply, but we actually can't guarantee that you're going to be allowed to step foot in the lab. It really depends on how the pandemic goes and the vaccination and all of that goes and how sort of university policy evolves. So that's definitely been a challenge. And so just as an example, we had 2 work studies who started in the lab this fall, and that program essentially wrapped up well in advance of when it normally would, just because we couldn't safely bring them in to continue their work.
Thank you. So hoping that the pandemic ends soon and we go back to normal procedures, after one applies for a position and are successful, you have to face an interview. So how can students really prepare for this interview? What are you looking for in the ideal student? So
I think the best thing that students can do is make sure they have an understanding of what the researcher is studying. You'd be surprised how many students show up and don't have the slightest idea that I work on macrophages, for example. It's kind of weird, but obviously that's not who you want to be if you're trying to make a good impression. We don't expect you to know our research inside and out. I mean, half the time I don't know my research inside and out, so I would expect someone applying to the lab to have that same grasp. But you should know the basics. You should have an idea of what the major projects are. And the other thing that I really look for is that the student has a clear idea of what they want to get from the position. So whether it's, they want to go to med school and they want to kind of understand a bit more about immunology, or they're maybe thinking of a research career and they're looking to get a foot in the door that way, thinking of an industry career and want to learn some relevant methods or whatever. That's, I think, is an important thing to communicate to someone that you're interviewing with, because not only does it help us understand better what you're looking for, but if we're now designing a program for you, a research program for you, we can try and keep those career goals in mind to make sure that that program, matches your interests and needs as best as it can. So you mentioned going into industry. And so if there was a student looking to go into industry, something different from what our conference focuses on, do you have any tips, or how is that program specialized for these kinds of people looking to go into industry? Yeah, so industry is a tough one because there's a lot of industry jobs and they're all very, very different. So, the main thing that we can focus on for a student interested in industry is, first of all, actually, maybe taking a step out of my lab, is there's a really good intern program that Western runs. And I've had a number of my students go through it, and a lot of them, they come, so usually you do that between your 3rd and your 4th year. So a lot of them, after finishing their 4th year, go right back to those companies into the beginning of a lifelong career type situation. So if you're really considering an industry job, that's a great place to start, is that internship program. At the level of a lab like mine, what we can do is we can try and focus on teaching you sort of a broad array of technical methods, and as well as data analysis, because knowing how to analyze data is really important for a lot of industry careers. And you can learn with one technique, but once you kind of understand the concept behind analysis, it's then easy to transition into other techniques. So even if you don't learn a particular technique you might need in industry, you've at least kind of learned how to think about data and critically analyze it. And by giving sort of as broad a base of methods as we can, that also means that there's at least a better chance that you'll have a technical expertise that an industry partner is looking for when you're starting out. But again, if you're thinking about industry, that internship program is a great place to start.
So what are some of your kind of opinions on academia versus industry, and how did you kind of determine what was the best fit for yourself?
That's that's a tough question. For me, I never really had a huge interest in going into industry. I really liked sort of pursuing my own questions. I was really fortunate, both in my PhD and in my postdoc, to have supervisors who really let me kind of find things I found interesting, and within obviously the confines of their program, sort of pursue those interests. And so I really enjoy that. I think that's one of the favorite parts of my job is, as long as I can convince a funding agency that something I want to look at is cool, I can then pursue that. Whereas, industry tends to be a lot more confined, because there you're trying to develop a product or complete a safety assessment or something like that. So it's maybe not as open and free as an academic position. And, a lot of people, I think that the teaching aspect of academia maybe is a little scary, and they don't really want to deal with that. Of course, there's just the job market, right? For every academic job, there's probably 50 industry jobs. So it can be a little easier to land an industry position to get your foot in the door there than it is in academia.
For students that are looking at kind of pursuing a future in potentially academia or even industry, and are going on to do kind of their master's and PhD or even kind of a postdoc, you mentioned you had some very strong supervisors and mentors kind of in your path. So what do you suggest kind of looking for in those people in those positions?
Yeah. So obviously the first thing is, when you're looking for someone to work for, it needs to be in an area you find interesting, right? Because it can be very hard to keep up motivation sometimes. Everyone, especially in your sort of PhD years, often hit points where it's just a real grind to get through, and you just have to put your nose down and get the work done. And it's easier to do that if you actually find what you're doing interesting than if it's a chore, right? It's hard either way, but it's easier to keep the motivation up, at least if you enjoy what you're studying. So that's a big part. Make sure you're going into a field you think is interesting. The other thing, when you're looking for a supervisor, it's always a conflict between trying to find sort of a world-renowned scientist whose name carries a lot of weight versus finding someone who is supportive of trainees. And that's not to say everyone who's really well known is not supportive. Lots of them are, but you need to make sure that you're going to get the supports you need from that person, and that can be very personal. What one person needs is very different from someone else. Some people are pretty independent, and the main thing they want is their supervisor to stay out of the way. Other people need very different types of supports from their supervisor. If you're someone who likes independence, you don't want a micromanager leaning over your shoulder, and if you need a little bit more support and stuff, you don't want someone who's sort of a hands-off, laissez-faire type person as a supervisor. And other than that, that's really the two big ones, right? Make sure you're a good personality match with your supervisor, and make sure you're doing something you find interesting.
Perfectly said. And you were talking about how, you need funding for all these research projects. And as an undergrad, you can really use NSERC as a valuable tool. So would you mind describing a bit more about what it is?
So NSERC is one of Canada's three major funding agencies, federal government funding agencies, I should say. And their focus is really on sort of all the science that isn't medicine. So they cover everything from, particle physics and engineering through to agricultural science, through to basic biology. And I'm sure I missed half a dozen fields in there that they cover as well. So NSERC has a undergraduate research program called the WSRA. And I apologize, I don't remember what that acronym stands for, but it's a summer studentship program. So it pays most of your stipend. Your supervisor does have to be NSERC funded, though, for you to qualify for that. Unless they've changed that rule this year. I'm not sure if they have or not, but in past years, that was always the case. And it essentially gets you in the lab over the summer. It's a chance to start doing research. You get paid, which is always nice. And it's a competitive application, right? Most, a lot of people apply for it, but there's only so many available. So if you get one, it also looks really good on your CV because it's, competitive national level funding for a student position. And so it's a great opportunity if you can get one to get into a lab and do some research. And it's nice because it is a program, like I said, that basically is available in any of the departments across sort of the science and technology parts of the university, including a lot of people in the med school, not just in the non-med school components of the university. one other summer student program on Western for science students, or at least medical science students, and that's the DURA program, which is the Dean's Undergraduate Research Summer Program, or whatever that acronym is. And that it's same idea. So it's a competitive application available to any student within the medical sciences program. Sorry, I should say with any supervisor in the medical sciences program. Covers a summer stipend. One of the things with the DURA program that's a little different from the USRA is they actually have sort of some student presentations and stuff like that. So it's not just research, but you get an opportunity to present your research to the other DURA recipients and their supervisors and labs. So it's a little bit more of a thing than just a stipend. You know, there's a few extra little pieces to it, which is kind of nice. And to the best of my recollection, those are the sort of major summer student funding streams that we have available to us.
And you also mentioned how, you know, the pandemic created some challenges. And I went to your lab, I think in the beginning of 2020, and we didn't know how the year was going to go. How did 2020 go for you? How did your lab change?
It was challenging. I had a new postdoc join the lab less than a month before we shut down. I had a new Ph.D. student join the lab in the middle of our shutdown. So trying to get them trained and up to speed was a challenge. And even now that the lab is partially reopened, it's still really challenging because the way we've always liked to train people is really one-on-one. You know, myself or my technician or an experienced person in the lab will take the new person and, you know, work with them through an assay a couple of times until they're ready to do it on their own. You can't do that if you have to social distance, and you can't do that if the lab is at 40% capacity or 60% capacity. And so it's made it really challenging to get people up to speed and to teach them some of the sort of methods that are unique to our lab that they wouldn't have had previous experience in. And then there's been other challenges that have really kind of made things a little more difficult. So we transitioned to online teaching. I'm sure you guys found that to be a whole ball of trouble, and it's just as difficult on the faculty end too. I teach a lab-based course, so I had to shoot a whole bunch of videos to demonstrate all these different methods, and trying to run the course online while trying to keep my lab afloat, while trying to do all the other things they make us do. It was a lot. And so that adds to the stress and the pressure quite a bit, having to deal with a lot of these things on the side, in addition to trying to keep the lab running and trying to get your new people trained and all that kind of stuff. And then some other things that have been problems too is just getting access to materials. Test tubes are a hot item right now. They are very difficult to get. We've been waiting for 6 weeks on some of those because the companies making them, they're all getting used for COVID testing, so it's hard for the rest of us to get them. And masks and gloves for a while were very difficult to come by, and you can't work in a lab safely without those right now. So there's been a lot of challenges, some of them a little more unexpected than others, but we've made it through, and I think we're closer to the end than the beginning, I hope, after sort of a bit of a hiatus there for a while.
So you mentioned some of the challenges you have while conducting your research. Have you experienced any challenges disseminating what your findings and just in general, the dissemination of information? Has that process changed?
A little bit. So, a lot of the conferences have moved online, which actually in some ways has helped us disseminate research. It's easier to attend an online conference in some ways than it is to travel somewhere to do it. The challenge of attending an online conference is actually sort of not trying to multitask and do other work stuff at the same time. But it also means, though, that, conferences that normally we wouldn't attend because they're on another continent or because they're maybe not as specialized as we might otherwise attend are now more easily accessible. And so we've, myself and some of my other lab members have been attending some of these online conferences. In terms of publications, it's been pretty normal, I would say. If anything, we're a little ahead of where we normally would be in the year, just because we're not in the lab as much, so it gives you a little more time to work on writing up and finishing up papers. So that part's been okay.
Well, you heard it here first from Dr. Heit. Our conference is going virtual, so there are many benefits to that as well. So We've gone over how 2020 looked like for you, but what does the future look like? What does 2021 have in store? Do you want to elaborate on some, key projects you're working on?
Yeah, so we have a small COVID project ongoing in the lab, trying to understand how COVID manipulates some of the ways that our immune system would normally detect a pathogen like COVID-19. So that's a project that we've put a fair amount of our effort and resources into, and hopefully that'll bear some fruit soon. And then our atherosclerosis project, we just had a large study published, but actually that project's really moved forward quickly. The Ph.D. student I'm working on that is very talented and very hardworking, and she's managed to get some experiments done much more quickly than I thought. And so I think we'll be seeing another paper coming out of that pretty quickly here as well. And actually, on the note of undergraduate researchers, contributing to that as an honor student in my lab, and today he was just showing me some really exciting data that took him all of last term to get the method to work, but now it's producing some really beautiful results. So that's an exciting project, and that's a method and an approach that was entirely developed and now being used by an undergraduate.
Well, that's super interesting. Thank you so much for coming onto the abstract to talk about, the opportunities and resources that undergraduates should be more aware of, as well as what it means to, be a scientist. And I know that you've done so many amazing work, and I'm so glad that you touched upon so many things that you did. And of course, what's going on in your amazing lab, because you're working on some amazing things as well.
Thank you for having me.